webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website, autism.org.
And so the topic today is the gestational influence on the etiology of autism. And this is an area of research that my lab is very involved in, and we think is really important, is understanding what is going on during that gestational period that might impact the development of the child. Excuse me, allergy season here. So autism has several suggested etiologies. I mean, environmental insults, certainly genetic predisposition, and this can mean direct genetic effects, or it can mean genetic predisposition that provides a susceptibility factor that then something else can come in, like the immune system, or some other factor might come into play. The gut bacterials, the microbiome is a big area of research. Infection during gestation is also something that is been looked at for quite a long time. I'm going to go through some of that today. Antibody antibodies is something else I'm going to go through today. It's something that has certainly gained some traction in the last couple of decades. And then when we say skewed, we mean sort of a dysregulation, or they're not the normal patterns of cytokines that we see, and there may be some increase in pro-inflammatory cytokines. And that also can go along with this infection model. So gestational immune dysregulation, what is this? There's epidemiologic evidence that implicates maternal infection as a risk factor, not necessarily with a specific agent, but more with the idea of inflammation relative to infection. So this is the idea that you have this sort of skewing of the immune response during pregnancy because of some insult. And this was really first described more in schizophrenia, and sort of the notion that there was a seasonal affectedness with schizophrenia, that potentially getting sick during certain times of the year when we get flu more often was associated with a schizophrenia outcome in the offspring. And so this is sort of the model that we use of infection and neurodevelopment, or the maternal immune activation model. And this is the idea that when an inflammatory event takes place, I'm so sorry, my mouse, an inflammatory event takes place, that the mother responds to that event as she should, right, to clear the infection. But unfortunately, in some individuals, and not all, this results in maybe a more active response to this infection, and that can then transfer factors such as IL-6, cytokines such as IL-6, to the fetal compartment. We're thinking, and this is a little bit difficult to do unless we use an animal model, but trying to understand what of these factors can cross the placenta, what may act on the placenta, because that is another area of active investigation. But all of these taken together can then increase the risk of altered neurodevelopment. And this is sort of the working maternal immune activation model. So infection, fever response is one of the key parts of this, not what the agent is, but how the mother responds to the agent. And I think that is the important takeaway from that. So not everybody who gets sick has a child with a neurodevelopmental disorder. I mean, that is, we know that because we all get sick during pregnancy. But it's that how you respond and how long that response lasts. So this suggests that impaired immune regulation on the behalf of the mom's immune system may reduce the ability to control that inflammation, so that instead of getting a quick up down, you end up with some sort of sustained inflammation. Or it is somebody who has other underlying things going on, such as, an autoimmune sequelae that they don't even realize that has increased inflammatory cytokine production as part of its pathology. So genetic susceptibility is going to be part of what controls this. Gestational timing is also thought to be very important when this occurs during gestation. The intensity of the response and how long it lasts, as well as potential additional postnatal events. And so these are all things that we think may be playing a role and that we're working to understand. So we wanted to look at the sort of the risk factors in the clinical population for autism and developmental delay without autism. And for this, we use the early markers of autism studies. And this is a Southern California-based study. It was a collection of banked serum samples taken between 15 to 19 weeks, sort of during the second trimester, for alpha fetoprotein testing. And for that screening program, somebody had the foresight to bank this, which we're grateful for. This is a study that we worked with Kaiser Research, my colleague Lisa Crowan at Kaiser Research with. And these were collected in 2000, 2003. And then if anybody has looked at the numbers recently, I'm sure a lot of you have, the increase during this time, it was 1 in 500 cases. We're now at 1 in 36. So this is an older population, right? And so understanding what was going on then and compared to the newer studies that we're doing may also help us understand what potentially is going on. This is the timeline and the time period that these samples were taken. So as I said, this is in the second trimester. We think the second trimester is an important period one of the reasons for that, this is where the immune system is probably most tightly regulated in the mother. And early in the first trimester, as the implantation and everything is going on, and sort of the early development of the fetus, the immune system of the mom is starting to be under tighter control, so you don't reject that fetus. Remember that fetus seen as a foreign body because it's half the paternal side. And so that is a really important part of what the immune system does, is tightly control the maternal response so that it doesn't reject it. The downside of that is we may not be able to fight infection ourselves quite as easily at the cellular level, which would be like viruses. So these are all things that are going on. I mean, we're trying to balance healthy pregnancy with healthy mom. So we did the study in the EMA, and we compared mothers of children with autism with no intellectual disability, and mothers of children with autism with intellectual disability, developmental delay without autism, and then a set of general population controls. And this part is a very important aspect because most of the studies that I work on here at Davis, we have a validated, typically developing population, meaning we know that the child does not have any developmental delay, does not have autism. This study, with these controls, were samples that they had never sought services in the state of California, and so some could have been missed. And I just say that because as I'm talking through, I'm going to present 2 studies from this population, and it's an important aspect as we go through the talks. So I think this is a very, very kind of busy table, but I think just if you look at the places that are red, that is where these particular cytokines, and these are chemokines down here, are differentially higher in the ASD population that we're looking at. So the GM-CSF is a growth factor. TNF-alpha, IL-1 alpha, and IL-1 beta are innate inflammatory cytokines, meaning they're part of your innate protection, which is still very active during pregnancy. IL-6 and interferon gamma are Th1 cytokines, Th2 cytokines, IL-4, that is what drives your allergy response. IL-10 is a regulatory cytokine. We have the inflammatory cytokine IL-17 that fights infection. And we go through, and these are the innate inflammatory chemokines, just to give you sort of a little background on what these are. And what we noticed is when we compared ASD with intellectual disability, that seemed to be the population that was driving this. They had higher inflammation during pregnancy compared to the general population, compared to So now these we have both have intellectual disability, one with autism, one without, and you can see there's a skewing toward inflammation. But the most important piece of this is that mothers of children with ASD plus ID had elevated inflammatory cytokines over those without intellectual disability in their offspring. So this is now separating this population of autism from each other, right? So intellectual disability seems to be more associated with inflammation in this second trimester period. So this profile separated them from all other groups, and it suggests a lack of selective immune regulation in these women. So meaning if they had some sort of, either they had an underlying condition that had yet to be diagnosed in terms of inflammation, or they had some kind of perturbation of the immune system during that period that they were taken. Because remember, this is just a one-time point cross-sectional period. And I'm going to sort of move, because this is newer data, from what is going on in the mom to the child's immune system at birth, just for a moment, because I think this is, they are, and we've got some studies going on right now showing the relatedness between what the mom's profile looks like and what the offspring's profile, the child's profile looks like as far as their immune system at birth. And I think this is a really exciting area for us. So we are working on this right now to, and that's upcoming data. But what we do have is that we use the newborn blood spots. We're in the state of California and several states in the US. Blood spots are collected at birth for newborn screening, and this is a fantastic program that we have access to. And we can measure what the immune markers at birth look like. Now, some are going to have come from the mom, and some are going to have come from the child. And so newborns are born, when we're born, we have a stronger innate immune response to fight infection, but our, what we call our adaptive immune response, meaning the ones that make antibodies and our T cells that fight infection, only start developing really postnatally with exposures, right? Because they have, this immune system has to be educated. And so what we did notice, and this is, I think, probably a surprise for a lot of people, is that newborns that were diagnosed with autism and delayed development and had lower levels of cytokines and chemokines at birth compared to those with typical development. And this was a really sort of an interesting idea, and some of the things that we're working on in the relationship between what happens in the mom and what happens in the offspring, maybe explaining some of this, and so stay tuned for that. But our exploratory analysis suggested that these, we had CTAC and MPIF1, which are both chemokines, and chemokines are factors that tell cells where to go. They're like a signal from one cell to another to move in this direction to fight infection. But these seem to be the strongest predictors of autism compared to the typically developing and developmental delay populations, respectively. The higher levels of CTAC were associated with a 60% decrease in the odds of ASD relative to TD. So this is a really important sort of fact, that if you had more of this chemokine, then you had a lower odds of having an ASD diagnosis. So they're protective. And the reason this may be is because during development, these chemotactic factors are really important in getting cells where they're supposed to go in the brain and throughout the body. So not just immune cells, but also neuronal cells, microglia, all use the chemotactic or chemokines to signal where they go during development. So that might be one reason. We also noted that higher levels of the chemokine MPIF1 were associated with a 138% increase in the odds of ASD relative to DD. So, meaning the ASD population separated from the developmental delay without autism based on the levels of this chemokine being too high. And I think we often refer to sort of the Goldilocks effect when we're talking about cytokines and chemokines, because those you need some to have healthy neurodevelopment, and it's when they're skewed that we end up with a pathology. And the other thing that we wanted to take a look at was the idea of the offspring sex. And I think this is something that has really come out in the last couple years, is that difference that we're just innate between males and females in how our immune system functions, what the profile is, and especially at birth. And I think this is it's a kind of a weird graphic to try to explain. And this is a huge number of cytokines and chemokines here. But we did notice that neonatal immune signatures differ by sex, regardless of what their neurodevelopmental outcome. So females actually are born with more of these sort of the chemokine factors, a little stronger innate immune system than males. There was only one chemokine, the migration inhibitory factor, MIF, is the only factor that was different for autism and developmental delay compared to controls, and that was more in the females, where the males, that's what the blue are the males. We're very traditional here, and the pink are the females. And the farther the bar is, the more they had, and the little stars mean they were significantly different. But you can just see that the females were significantly higher in several of these. And this is, I think, one of the reasons that we looked at this is because when we are trying to understand what is sort of typical and what may be pathogenic, we really need to understand what we're working with. And if we're treating males and females as the same, then we're really not understanding things correctly, right? We're getting misinformation. So by looking at this and saying, no, we need to really look at them as separate populations biologically as well as behaviorally. So that is really was the takeaway from this, that control males had higher BIF than females, where DD females were higher, AU and DD females were higher than the autism males. And so that was this bar right here, just for one chemokine. So that was the only one that distinguished them by their behavioral outcome, and the rest were based just solely on the sex of the child. So I'm going to switch gears a little bit. I sort of did with the cytokine chemokine profiling and what was going on during development from that aspect. And now I'm going to sort of change topic a little bit to another type of immune dysregulation during pregnancy. And this is maternal autoantibodies that target proteins in the developing fetal brain. Some of you may have heard me speak on this before, and we keep I try to keep updating where we are on this research and what is coming up. So just to give everybody the background, maternal autoantibody related autism is a subtype of autism. And I think we need to be very clear about that. It's about 20% of cases that women have antibodies that are reactive against proteins in the developing brain. During gestation, IgG crosses the placenta to protect your fetus from everything you've been exposed to, because the offspring, the fetus does not have its own functional immune system at this time point. And so everything you've been exposed to that you've made IgG antibodies to go in and passively protect that fetus. Those last till about 9 months postnatally. But unfortunately, when we have antibodies that recognize self-proteins, those also cross along with the healthy antibodies. Those can bind to the neuroprogenitor cells, and these are cells that become neurons, and they can affect their migration, their maturation, what they look like, and their function. And so we look at this through our animal models. And unfortunately, this then leads to changes in the way the brain develops. We see enlargement in brain, and we see ASD behaviors as a result of this. So I will go through this a little bit more detail. We identified this is not a simple one antibody. It would have been nice had it been, but it is actually combinations of 2 or more to 8 different proteins. Each of these proteins have a different function along the path of how the neurons develop in the brain. And so this is important to know, because you'll never hit this part right here, because this all happens before antibodies cross the placenta. They don't start that crossing the placenta, or they sort of start hitting their peak around day 110 postnatally. But as from the very early differentiation, each of these proteins that are recognized by these antibodies have a different role, with the most mature being CRIMP1 and CRIMP2 toward the end of neuron development. And the axon is the part that is the signaling part of the neuron. So the effect is additive, meaning if you have just one, it's not enough. You may see some changes, but you certainly won't cross the threshold for an autism diagnosis. But when you start having two or more, especially CRIMP1 and GDA, CRIMP1, CRIMP2, STIP1, and NSE, those are my top three. And I'll tell you how we arrived at that. We actually have created an assay, and this assay, in full disclosure, we have developed a company to try to get this to market. That is a very long, slow process, as research is, but that is even longer and slower, unfortunately, because we want to make sure we get it right. And so what we did is we developed an assay where we took each of these proteins and coated a plate. This is not anything you need to know specifically, but what we do is we measure how much of these antibodies bind to each one of these antigens or proteins, which are the targets of these autoantibodies. And there's a long process we train to decide what are the patterns, and then we have to do a validation to make sure we're correct and that we're correctly identifying it. There are 4 statisticians shown here, and that is what it took to actually create what we now have as our MAR antibody test. When we did this, we found, and this is just, and I should this box, yes. So these are the top patterns that we found that were 100% specific for autism, meaning we do not see them in the typically developing population. This adds up to, if you add everything, it's about 20% of the population was positive in this study. But when we look at odds ratios over here, this is because we don't have, we have a high number of samples. We had 450 autism and about 375 typically developing. This would have probably been a higher number had we had more, but this tells you that we have 31 times more likely, if you have this pattern, that you will have a child with autism. And then these are just, so that's what the odds ratios tell you. And even for those patterns that are not 100% specific, that's what this precision number is here, we still have 7 or 8 times higher odds of having a child with autism if you're positive. So as we are going forward and developing this assay, those are the numbers that we look at to try to be very specific. And I see we have a hand raised. Are we going to do that? I'm happy to pause and answer questions on that part. Okay. So don't forget your questions, because I love answering questions. So the other thing that we noticed is if the antibody that was present was CRIMP1 in anyone who had a child with autism, it increased the odds of severity. So the 2.3, so if we have an ADOS severity scale of 1 to 10, if you had a 6, you would go to an 8.3 on that scale, meaning that just that antibody present is enough to increase the severity of the child's autism. And so this is, now we have seen this in more than one study. So I think we are very, very secure in our findings on this, that this means that not only do we see a specificity for autism with these antibodies, but we also see phenotypic differences. And I'm going to, apparently I said it already, but I'm going to go through that in this other study that we did. So remember I talked about the early markers of autism study that I showed you, the cytokine and chemokine levels were different. Now we also looked at these autoantibodies in that same population. And my student now is marrying those two findings, and we will report on that sometime later this year. But we did the same study, and these are now, the women are pregnant. We don't intend to ever test during pregnancy, but testing in the research side during pregnancy gives us an idea that if the antibodies are present and the outcome of the child is autism, that they were there before the diagnosis versus the first study I showed you, which was a postnatal study, and the samples were taken at the time of the child's diagnosis. Now we know if they're on board that they are like they were present during the development of that child. But we also wanted to validate the patterns that we previously described. And so what we did is we did the same ELISA, and this time we had patterns ahead of time that we were testing, right? So now what we were doing is taking those original patterns that I described in the charge study, and we applied them to this population. One of the interesting things was when CRIMP1 GDA in the previous study was the primary most abundant pattern, now CRIMP1, CRIMP2 is, and NSE STIP1, which was also way up there, is down here. So we do see differences. Now, this is a Southern California population. The previous study was a Northern California population. I've done other studies where we've looked at CHOP in Philly, Arkansas. The rates and the percentages of positives and the patterns they differ. And I think this is a really important thing. If we're going to use this assay someday, we need to get a huge population to test from all over, different ethnic backgrounds, geographic backgrounds, so that we can really truly assess what the percentage is in the population. But that is to say that if you get reactivity to a single protein that is very nonspecific, regardless of what the outcome of the child is. But when the reactivity to a MAR positive ASD it's highly associated with ASD and not developmental delay without autism. And that is a population that we had in this study that we did not have in the first study, which was the developmental delay without autism. We did see a little bit of leakiness here where we have a couple, and this is actually one person for these three. And remember when I said that the general population controls here were not ascertained to have autism or not by a regional center, unlike the ASD population, which were seen by a regional center. So, but we did validate the patterns. We see a little bit of a different representation of the patterns in the population. And what we also noticed was that there were patterns, especially that had CRIMP1 in them again, that had a higher rate of intellectual disability when in this population versus the general population controls. We saw a greater representation with the intellectual disability versus the non-intellectual disability associated with autism. And then conversely, we actually saw, we also saw that there were some patterns that were more ASD no ID versus GP. Now, CRIMP1 is with STIP1 down here, and it was seen more in the ASD no ID population than so there. If you look over here, these are the odd. This is the separation between ASD with ID and ASD with no ID. So to us, that was interesting, and I think just in the practice and usefulness of this, if we can understand sort of the type of autism that these predict, then we can target those, sort of in terms of therapy and treatment, you can target your interventions to be more useful instead of just throwing everything at a child. So I think this is one of our the focus of what we want to do going forward is really understanding the behavior nuances in these kids, because our goal is to help get the most effective interventions sooner. So just the conclusions for that, the clinical studies that I talked about, we found that reactivity to any of the MAR patterns was highly accurate in their ability to predict autism risk in both the studies that we did. Reactivity to CRIMP1 and CRIMP2 was associated with more severe ASD with an intellectual disability co-occurrence. So remember, CRIMP1 and CRIMP2 were at the end of neuron development. So now we're at that part where we're getting a we're potentially affecting the function of that axon. Some patterns tend to be associated with ID and with no ID. So that hopefully will, as I said, will help us provide insightful information about the phenotype or the type of autism, because we know autism is it's such a complex, disorder that we want to understand the nuances, because that is when we are more effective with the intervention services. And we also see, as I said, we show different distribution depending on the geographical differences in the two study populations. The other thing that was different, because we didn't see quite as high a percentage in the EMA study, is that was taken, remember, at 2000 when the incidence of autism was lower, versus the CHARGE study is a newer study, more recent, in sort of around like 2012, 2015. So the incidence had increased at that time. So we actually are seeing sort of with the increased incidence, we're seeing an increase in MAR as well. So that is the sort of what we've done clinically, and we continue absolutely clinically to work on this. But one of the important questions for us was, are they pathogenic? And by that, I mean, do they cause the antibodies themselves cause the change, or are they just a biomarker for something that has happened? And that is a very important distinction. And which of them? Is it all of them are all of them pathogenic, or just some of them? And so these are questions that we're trying to answer through our animal models. So we use animal models to help us understand what these antibodies are doing. We determine, do they localize to the brain? We found that they do. And so we create clinically, and then we created clinically relevant, meaning we took the pieces of the protein that the humans react to, so the moms react to, and we created those pieces and put them in an animal so that the animals would make an antibody response that looked just like the humans. And that's a very important part of what we're doing, because some of those pieces of protein are very specifically reacted to by MAR-ASD moms who have children with autism versus just the general population reacting to a part of the protein. And that's a very complicated concept, but that is one of the things that we're working on. So what does our model look like? We take those, what we call MAR-ASD peptides, meaning these are specifically reacted to by women who have a child with autism that are reacting to those proteins that I described. We inject the DAM. We've done this in rats and mice. Then now, the difference between this and the passive transfer, where we took a human antibody and put them into the animals, which were our first models, now the DAM is making these antibodies. And two things. One is a higher titer that lasts longer, and it's there for the entire incubation, right? The entire pregnancy. And the other important part is we do this before we ever breed the animals. So there's no inflammation at the time of breeding. And I think sometimes our model gets a little bit confused with the maternal immune activation model, but we've tested this. There are no there's no elevated cytokine during the time of breeding, because we've done this ahead of time, let everything kind of calm down before we even breed the animals. So then we look at the offspring on several measures. We do behavior. We did imaging of the brain. We're looking at them under the microscope. Where are these antibodies in the brain? And now we're moving on to culturing cells to see if we can impact, to understand how the antibodies are getting into the cells that they're getting into. So both our mice and rats display altered social behavior and self-grooming behaviors, meaning, so this is the mouse study over here. These are they are reduced in some of their social behavior measures and increased in self-grooming models. In the rats, we see, oh my goodness, Judy. We see that, and this is now published. I should have fixed that. But now we see that rats, and one of the reasons we moved to rats, it is so fun to watch them. They play, they roll around. Mice have social behaviors. Rats are incredibly social. They also have some prefrontal cortex, so they're a little higher. They've got more sort of more refined behaviors that we can look at. But if you just look at how much they play, they're well below. And juvenile is very This is the active time where they're just rolling around the cage together. And you can see that just that behavior decreases over time, but the MAR offspring are always lower than the control offspring. And rats, unlike mice, which do a lot of self-grooming, rats typically do not. But in the case of our rats, especially as adults, they do much more self-grooming and sort of repetitive type behaviors. So this is something that was really interesting. The models are very similar to each other. And I will play a video of, and you can see which is the control mouse. So this is the sociability, and this is juvenile animals. So we've put a stimulus mouse that they've never met. That's our control animal. We've put a stimulus mouse in to see if they have, their sociability, and they stay together. There's, sniffing and crawling. I would say the stimulus mouse doesn't look very social, but our control animal does. And so we just showed that was their stimulus mouse is self-grooming a little bit. Mice self-groom a lot more. But now you can see the MAR-ASD mouse, it's very definitive. And the stimulus mouse is trying very hard to engage the MAR-ASD mouse, and that animal is not interested and wants to self-groom and actually actively avoids this social interaction. So they go across the cage so that they don't have to interact. And the other mouse will come and try again, and this just keeps happening. And we see the same thing in our rats. The rats, you can probably tell. Now, this is where you'll get a really good idea of how much they play and how much they interact. They roll around together. They're just, and you can see how fast this, the poor people who have to score these videos constantly have to stop them. A little mounting behavior, but they are very, very social. And the control rat is the one with the dark mark on it, and the stimulus rat. So now here's the MAR-ASD rat is in there, and you'll see the stimulus rat is trying, but the MAR-ASD rat is not very interested and kind of just does not reciprocate that attention. And eventually, this is different for the rats do this where the mice don't. The stimulus rat kind of gives up, and the other one just ends up just licking the bottom of the cage. So you can see one rat is trying and the other rat is not engaging in that social behavior. We've also looked at these animals have altered brain metabolite levels. We've done structural imaging studies where they're also different. And these are just telling us that we have altered the sort of connectivity and the way the brain is functioning with these antibodies. We also see that there is altered brain volume. So we have decreased communication, decreased social behavior, increased repetitive behaviors in these rats. And we see the change in brain volume where the males are low and the females are high. And that is something that's a little bit different than we saw in the human studies and in our monkey studies. But we saw the same thing in both the mouse and rat. This is the rat model. But we see there are places where the males are very different, and then the females are very different in some regions, and then there's difference in both. That is the same, midbrain and cerebellum. So I think we're still continuing to work on this. So this was this paper just came out in Molecular Psychiatry. So gestational development and immune dysregulation may contribute to risk of altered neurodevelopment. This may be an underlying mechanism behind increased risk due to infections during pregnancy, but the specificity for autism I don't think is clear at this time. I think because we're using the same model to look at schizophrenia, so it may be more a case of altered neurodevelopment and how intense it is, the timing, all those may determine whether it's schizophrenia or autism, and that is something that we just need to explore further. In vitro studies demonstrate the effect of inflammatory cytokines on neuron health and development. I didn't show that slide, but one of my graduate students showed that if you put the same array of inflammatory cytokines that I talked about in the first part of the talk onto developing neurons, it changes the way they develop. So there is an effect of these cytokines on neuron development. The question is, just how do we, distinguish that? And so antibodies do localize to the brain of the developing animal. We've shown that in both monkey and rodent models. We see enlarged brains in humans, mice, and monkeys and rats. However, there seems to be a sex difference in the rats and in the mice with respect to these enlarged brain regions. And it's more complicated. There's not a difference in their behavior, but the parts of the brains that are either larger or smaller are different, whether they're male or female. So stay tuned as we work on that one. And then mouse and rat models capture all three domains of ASD, and this is our clinical, meaning they're making the same antibodies as the humans. In the social communication and repetitive behavior deficits, they don't have other, we don't see anxiety, we don't see other things that may be associated with autism in this particular model. So this definitely has its own phenotype. And then we know that, as I said, that they alter neurodevelopment, including the total cerebral volume in the several of the studies we've done, which just sort of demonstrates that there are structural effects of these antibodies, that it's changing, physically changing the way the brain develops. Oopsie, let me go back. And I have an incredible group that I work with. There are many more listed here, but Melissa Bauman does the monkey and rodent behavior. She's a neuroscientist in behavior, specialty. Paul Ashwood is also an immunologist. Many of you may know who Paul is. Dr. Lisa Crone is an epidemiologist that specializes in autism at Kaiser Research. Matt did the rats. Ale did the human clinical studies. Danny did the blood spots. Jana has taken over the rats from Matt. So we're continuing to look at the ones I showed you were one pattern, and now we are breaking it down into those patterns I showed you that are the clinical patterns, the top 3 patterns. And Jana is carrying that on, and Chelsea's working on our schizophrenia model. So with that, I will take questions. I will go to my first slide, and then we can do some questions.
Great, thank you so much for the presentation. We've got lots of Q&A here. So we typically, I want to explain about the hand raising. It just, sometimes when we do that in the middle, it can be we'll get off track a little bit. So we'd love it when people type in their questions, then we can answer them at the end. So this first one, Judy, is about schizophrenia. They're asking if in any of the research that you've seen or any of the research you've done, that correlation has come up, if you've seen that specifically.
With ASD and schizophrenia, I think that is a really interesting question. And I think the way we're looking at that is they both can have these very early immune development issues, and how they go down the path of autism versus schizophrenia, or maybe some of the overlapping, probably more in the prodromal side of schizophrenia, because psychotic break is less common in autism. But certainly there are some, anxiety for sure is something that you see in both. So I think it'll be really interesting to understand that overlap between those two in the future.
So more research to come on that one.
Yep.
Okay. Yeah.
Lots more.
Yep. Okay. Did any of your studies take into account whether the mother had a diagnosis of autism herself? So was that some of the data you collected?
And I have not done that. So I don't know in terms of if the mother had ever been diagnosed. In all of the studies I talked about, at least or in charge, the mothers had not been diagnosed with autism. So, but I think that is an incredibly interesting question. I mean, there are a lot of ways to get to autism, right? So this is just one of them. But that is a really interesting question that I would love to address.
So this is a question about miscarriage and correlation here. If the mother has suffered a number of miscarriages during the first trimester previously, might that be an indicator that the immune system is affecting the fetus? And in that case, with the 9th pregnancy then ends up with a diagnosis of autism, is it just coincidental? I mean, I think based on your environmental research, you'd suggest not, but have you done any research or seen research specifically addressing that?
I think certainly miscarriage and autism has been looked at, right? The number of pregnancies time between pregnancies, miscarriage rate, which suggests absolutely that there may be immune dysregulation at play, especially if you have multiple ones. I think for the work that we're doing, I have not gotten a lot. We don't have a lot of participants in the studies I've done that we have either the data for or that have experienced that. But I think it's a really interesting question because we always understanding the underlying cause of a miscarriage. I think we typically did not look at the immune system as the factor that might be driving that previously, but there are studies like Stanford does, both they have a big study on not just miscarriage, but also prematurity and how that impacts things and how the immune system impacts both of those things. So there are people doing that work.
All right. This next question is about dads. So based on this information, does it suggest that dad has no influence on a child having autism?
I think that,
that's a loaded question.
That is a very loaded question.
Yeah.
For the maternal autoantibody model, it is unlikely. However, what we have not done and what the study that needs to be done that we need to get funded to do is where the overlap with genetics and these autoantibodies, where that intersection is. Because you can imagine that the autoantibodies set the stage for some things, but if you have paternal or the dad's influence genetics on that, it may have a completely different outcome, right? It may exacerbate that. It may be a different phenotype. So I think that is an excellent question, and I would love to do the study to understand that.
All right. Here's a question. This person says, excellent presentation. As an immunologist, I agree with so much of what you've expressed. Thank you so much. I would like to know if studying markers such as TNF, BDNF, S100B, or CKBB would help us evaluate neurocerebral dysregulation. So that's a big question, but they asked.
So we've done work certainly on TNF and BDNF, and a couple of people did more in the newborn blood spot for the BDNF. TNF was one of the markers that I talked about in the maternal inflammation. And I think in terms of looking at them specifically, I try not to look at just a single factor or one or two because they are not acting alone ever. And when one is up, a whole host of other ones are up. And so I think it's really understanding what the whole sort of milieu of those factors are throughout pregnancy, which is a study that we've got going now with Lisa Crone. Our impact study is understanding over time what these look like. And we're including in that metabolic markers because sort of a metabolic dysregulation is also an inflammatory event. Oops, my mouse is like a continuous mouse. And if I touch it, just keep my hands off of it, it will move my slides.
Okay. So the next question, do you have any perspective or data regarding bipolar disorder and autism as related to immune dysregulation? If not, any preliminary thoughts?
That is also an excellent question. I think some of the data on schizophrenia, bipolar, and autism from the genetic side in the brain, the postmortem brain, suggests that they are quite different in terms of the markers that are related to immune function that are seen genetically. And that was work done by Dan Geschwin, that he's done quite a bit on that. So I have less bipolar disorder in the projects I'm doing on immunity and autism, a little bit more in our Conti Center. We do schizophrenia, and working with Cam Carter and Tyler Lesh is looking at schizophrenia versus bipolar and the immune, but that is not in neurodevelopment or brain. That is really in real time at the time, that these patients are being treated. So I think, that our early data suggests that there is a difference. It sort of stands alone from the other two disorders. But we'll have to stay tuned on what comes out from that.
This next question is about pregnancy and chemotherapy. So was any data collected about that, about parents who had cancer diagnosis or who had actually gone through chemotherapy and then had this, later had a child with autism?
Wow, that is a tough one. And it is always going to depend on the type of chemotherapy, right? There are so many, and they have different effects on different systems. And so I think that's a bigger question that we can answer right now. I'm not aware of any studies that have addressed that.
And we've got a number of questions along those lines, questions about IVF and different immune response, these sort of just a lot of medical procedures, how those may relate to or correlate with the eventual diagnosis of autism and how that connects with these different maternal autoantibodies. So interesting thoughts.
No, and I think, just autoimmunity in general during pregnancy, right? If how it's being managed during pregnancy. There are a number, the reason I got into this is because there are a number of autoimmune diseases that have downstream effects on the offspring, like congenital heart block and SLE, right? Thrombocytopenia, thrombocytopenia, God, Judy, which is really highly impacts the offspring viability. So there's a number of autoimmune disorders that women have that can impact the outcome of their child. Autism may be one of those. I think that is something that people are looking at more now than we used to, because it used to be more about the sort of physiologic effects.
So this next question is about subsequent pregnancies. So could the antibodies be getting stronger with each pregnancy? Did any of the dams that you used, did you do multiple generations? Or?
And we are, yeah. So the because we artificially kind of created and broke tolerance to those, I would say the titer lasts a long time. That I can tell you. And each pregnancy that we do, and in the human studies, we're doing the MARBLE study right now, looking at the high risk, they've already had a child with autism, and for some of them we have, prior to that pregnancy, we have a sample that we can look at. It doesn't seem to change. It seems pretty stable. We don't know what causes these antibodies to be formed, right? We don't know that pregnancy has anything to do with it. I think that is a very important study to do. I think it's assumed because we think it acts during pregnancy, that pregnancy has something to do with the generation of these antibodies, but we don't know that at all. And so those are all important questions, and sort of the multiple pregnancy idea. I do know if they've had it in one pregnancy and their child is positive, and they have it in another pregnancy and their child will also be positive. Their child will also have autism, just from the limited number that we've done.
Got it. Okay. That's important to know, and interesting about the research you're doing and the work that is being done to make that testing available so people can get that done ahead of time. As I said, there are a lot of questions here about different medical procedures and different medical treatments and the eventual diagnosis. Do you have advice for parents? I think, somebody's contemplating pregnancy. I know you can't give medical advice, but
no, that would be against the law.
Right. But thoughts about just how to approach the research, or how to speak with their clinicians about this different information.
So I think one of the, and I'm sort of scrolling through the Q&A, and thank you to everybody who sent me notes. But I think there are some things that, and I kind of landed on the one here with the implications of maternal lifestyle to positively, and I think gestational diabetes, sort of metabolic disorder, obesity are factors that could be potentially modified, right, for a healthier pregnancy, because those are linked to increased inflammation, and in one of the studies we did, to these autoantibodies. So I think that is something that we're we're looking at in the impact study, and we'll have a lot more data to support that But we try to always look at not just things that you can't really you can't help it if you make autoantibodies. There's nothing you can do about that. But in terms of inflammation during pregnancy, like a non-inflammatory diets, definitely, it turns out that, increased obesity is a factor in a couple of studies that we're doing. So those are things that potentially that healthier sort of pregnancy from that side, from the maternal health side, is important.